Good morning. I'm happy to open this second session. My name is Lee Krug. I work at Bristol Myers Squibb on immunotherapy for lung and head and neck cancers. And I'm happy to moderate this next session, which will be on the challenges with preclinical models for immunotherapies. And the premise for this session is that mouse models, which of course are created with various degrees of immunodeficiencies in order to provide the opportunities for xenografting, have inherent challenges when trying to study immunotherapies. And so each of the speakers is going to touch on the various different immunotherapy topics that we've already started to hear about, the immune modulators, the adoptive cell transfer, and the vaccines. And then we're also going to hear about the needs for or how to use these models when trying to initiate clinical trials and how to adapt that. And so we'll have some discussion about that. I'll introduce all of the speakers as we start. And the first one that we have is Alex Snyder. Alex is an assistant attending physician at Memorial Sloan Kettering. She has done a lot of work in the area of immunotherapy. During her fellowship training, she worked in Jed Wolchuk's lab and was the lead author on the New England Journal paper on the using mutation burden and neoantigens as a way of eliciting responses in patients with melanoma. She now works on the gynecologic service there. And because of various different scheduling challenges that we had, actually Alex is going to be joining us by teleconference. So what we'll do is she'll give her presentation. Hopefully we won't have any technical glitches. And then what we'll do is we'll take questions for her talk immediately after that. With the other speakers, we'll hold the questions until the end, and then we'll have a panel discussion for that. Okay, so Alex, without further ado, please go ahead.
Thank you, Lee, for your kind introduction. And if there's any issue hearing me, please just interrupt, and so we can sort it out. As Lee said, my name is Alex Snyder, and today I'm going to speak about the limitations as well as the benefits of preclinical models as they relate to immune modulators. Next slide. So in this talk today, I'm going to speak to three main points. The first is that the proof of principle of the importance of a given pathway can be modeled in mice. Next slide. The extremes of a toxicity profile can also be gathered from knockout models, although the details generally cannot. And finally, predicting a specific disease application is generally not realistic with mouse models, especially with genetically engineered mouse models where the genetic heterogeneity and complexity is limited. Next slide. So today, after a brief background, I'll talk about some of the pros and cons of implantable versus spontaneous immunocompetent models, followed by some of the principal advantages of preclinical models and then their major limitations. Next slide. So let's start with some background. Next slide. As you all well know, William Coley is considered by many to be the father of tumor immunology. His injection of Coley's toxins, a mixture of bacterial cultures, was found to induce tumor regression in a subset of patients. Could you advance? In the early 20th century, the use of inbred or syngeneic mice allowed scientists to engraft syngeneic tumors, i.e. tumors from mice of the same genetic background, and this could lead to immune-mediated rejection. This moment was the beginning of the concept of immune surveillance. Advance. McFarland Burnett and Lewis Thomas first proposed that the immune system recognizes and monitors tumors. While Stutman's data showed that chemically induced tumors didn't grow faster in immune-deficient mice, it turned out later that when comparing profoundly immunodeficient mice to immune-competent mice, chemically induced tumors were indeed rejected by immune-competent mice, and this has been beautifully shown over the years by Bob Schreiber and others. These preclinical data laid the foundation for the subsequent development of immunotherapies, which again, as you all know, has really accelerated over the past decade. Next slide. If you look at this beautiful review from Drew Pardal, we see a view of some of the many receptors and ligands that lead to a dampening or enhancement of the anti-tumor response of T cells, and thus represent targets for checkpoint blockade or agonism. Essentially, all interactions are now being targeted either in open clinical studies designated by red ovals or in preclinical or soon-to-be clinical studies in orange, and moreover, not every target is even shown here. Next slide. So with that background being said, let's move on to some of the pros and cons of the different models. Next slide. For decades, anti-cancer therapies have been tested using xenografts in immunodeficient mice, as Lee said. These are still being used to test targeted therapies, but are clearly not sufficient for testing hypotheses related to our therapies. While this concept may seem obvious to this audience, I still not infrequently find myself in the position of explaining why PDXs can't be used, at least until humanized mice are common, cheaper, and up and running to test immunotherapies. With that said, I'll move on to discuss some of the pros and cons of implantable versus spontaneous models. I'm not going to talk about humanized models today, as my feeling is that topic represents a talk unto itself, and these have not yet been used to make the major discoveries in the checkpoint blockade field. So implantable models, as you see here, have features such as rapid and relatively reliable tumor growth. They're available for multiple tumor types. We can intervene upon the cell lines before they're implanted. They can be implanted orthotopically, i.e. into the organ of tumor origin, or subcutaneously, in which case they can be easily monitored. And they're homogeneous, where in this situation there is some advantage, which is that we can use syngeneic lines, and the experimental results are somewhat more homogeneous and thus interpretable. On the right-hand side, if you'll advance, I use the term spontaneous models to here describe two very different classes of model. First, carcinogen-induced, and second, genetically engineered models, some of the contrasts of which I'll address shortly. So spontaneous models tend to exhibit greater heterogeneity, which in some ways is an advantage because this is how human tumors behave, but in other ways is a disadvantage. For carcinogen-induced tumors, the response and escape may be more similar to human tumors. Bob Schreiber's work has relied greatly on this principle. In addition, many but not all spontaneous models exhibit organ-specific tumor development. Next slide. On the other hand, implantable models do not recapitulate the rate or context of human tumor growth. Typically, cells are implanted and then treated within days of taking. This may translate into different immune interactions taking place systemically and in the tumor microenvironment. Advance, please. The spontaneous models, particularly the genetically engineered ones, on the other hand, take time. A single experiment can take up to 6 to 12 months. The greater variability in phenotype can make results more difficult to interpret. Importantly, in many genetically engineered models, mutations are continuously present from birth. These mice may therefore be tolerant to their tumors, and certain immunotherapies may thus be less effective or more difficult to test. Next slide. The following two examples of spontaneous models of sarcoma illustrate some of the issues highlighted in the previous slide. As you know, RAG2 deficiency prevents both T and B lymphocyte development. On the left-hand side, Tyler Jax's group used human soft tissue sarcomagenesis driven by a Cre-Lox-P regulated expression of oncogenic KRAS and deletion of P53. Sarcomas were induced in either immune-competent KRAS P53 mice, which are KP on the left, or lymphocyte-deficient KRAS P53 RAG knockout mice. The color red or blue denotes each individual tumor, and dashed lines represent those tumors transplanted into RAG2 knockout mice. So you can see here that these tumors, which essentially had only two tumor-driving genetic abnormalities, grew at the same rate in both immune-competent and immune-deficient mice, irrespective of whether they originally grew in an immune-competent or immunodeficient mice. Advance, please. However, similar experiments done with a different model of sarcoma show a different outcome. So in this study out of Schreiber's lab, they looked at a carcinogen-induced D42M1 cell line, which has over 2,800 mutations, One of these, called spectrum beta 2, generates a rejection antigen, so that when that clone is engrafted into syngeneic mice, it is rejected, as denoted by the downward sloping line. In contrast, control cells with triangles or cells transduced with wild-type spectrum beta 2 in squares grow unabated. I show these two examples to illustrate the point that we learn a tremendous amount from these, albeit imperfect, models. These models teach us about immunoediting, but do not tell us specifically what to do with human sarcomas. In summary, we can translate the concepts, but not the precise details from these mouse models. Next slide. In this second example, the same gene, GRP1, is altered to generate two slightly but importantly different models of melanoma. In the first, the mutation leads to mice that exhibited uninhibited melanocyte growth, but the tumors are non-invasive. The mouse that says control is the one that's what the mice look like with this deficiency. In the second model, del-TG3, there's a deletion within an intron of GRP1, and in this model, mice develop invasive disease. When you treat mice with either type of disease with a tumor-associated vaccine, you see that the GRM1 mice are protected, but the deletion TG3 mice are not. This example demonstrates that this choice of a model and the evaluation of any intervention in several models is critical to the modeling of any new therapy. Next slide. So what about principal advantages of preclinical models? Next slide. Although the topic for this talk is the limitations of mouse models, the following two examples will show how central they have been to fundamental discoveries that led to effective immunotherapies. To elucidate the in vivo function of CTLA-4, in a seminal paper, Bluestone and colleagues generated CTLA-4 deficient mice. These mice rapidly develop lymphoproliferative disease with multi-organ lymphocyte infiltration and tissue destruction, with particularly severe myocarditis and pancreatitis, and die by 3 to 4 weeks of age. In the left panel, we see wild-type mice's heart and pancreas as compared to their heavily infiltrated counterparts in the CTLA-4 knockout mice. Advance, please. In the second panel, a B7 negative colon cancer line is rejected from BALB/C mice treated with anti-CTLA-4, but not control or anti-CD28. Amazingly, within 10 years of these studies, ipilimumab was doing its job in humans. Next slide. In this second, again, seminal example, we see how in the leftmost panel, BALB/C mice with a myeloma model treated with anti-PD-L1 exhibited delayed tumor growth, as shown by the open squares. In the right-hand panel, both the myeloma line and B16 melanoma engrafted cells grew more slowly in PD-1 knockout mice, shown by black circles. Advance, please. Molecules such as 401BB, OX40, GITR, ICOS, LAG3, TIM3, and others have all been developed based on the use of mouse models and are now being tested in humans. Next slide. Now that these first couple of breakthroughs have led to human success, one of the biggest challenges I think we face as a field is how to combine checkpoint blockade therapies with each other, with other modalities, and in which cancers. In the left-hand panel, in a study out of Andy Min's lab, C4 denotes anti-CTLA-4 and P1 anti-PD-1. The top survival curve shows how the combination of radiation and CTLA-4 blockade is better than either intervention alone. The lower panel shows how radiation together with blockade of both checkpoints provides the best outcomes. The paper goes on to dissect the mechanism for this. You could advance, please. On the right-hand side, in studies also out of UPenn, the authors hypothesized that failed immune recognition or poor T-cell priming underlies weak clinical response to checkpoint blockade in pancreatic cancer. On the top here, they used cell lines from KPC mice, i.e. these are cell lines from genetically engineered mice that were subcutaneously implanted. When they treated with anti-CTLA-4 or anti-PD-1 alone or together, these were essentially ineffective. Advance, please. However, when combined with gemcitabine, nab-paclitaxel, and CD40 agonism, they see tumor growth stops and survival increases, if you look at the far right purple line. Advance, please. In the bottom panel, they treat KPC mice with spontaneously arising tumors, and benefit is still seen, that the survival curve is still off to the right, but it's less dramatic than with the transplantable model. Of course, these are just a couple of examples. There are very many others in the literature of successful combinations, including but certainly not limited to checkpoint targets, combination of checkpoint with GCSF, oncolytic viruses, vaccines, and other therapies. Next slide. The other important issue that the preclinical models allow us to address are related questions. Next slide. So certain questions are virtually impossible to ask prospectively in humans. In 2 recently published studies out of the Gajewski and Zitvogel labs, the impact of the mouse microbiome, gut microbiome specifically, on checkpoint blockade is studied. In this slide, Gajewski shows that the administration of JAK's feces alone, i.e. feces from mice raised in Jackson labs, to TAC mice, retards tumor growth and does so even more so when combined with anti-PD-L1, as shown by the dark black line at the bottom. Advance, please. In Zitvogel's group's paper, she shows that those mice who live in germ-free facilities, shown in panel B, or who are given broad-spectrum antibiotics, shown in panel C, lose their response to anti-CTLA-4 relative to normally housed mice, here abbreviated SPF, which stands for specific pathogen-free conditions. Eric Paymer and Jed Wolchek have recently shown the importance of gut microbiota composition to the occurrence of colitis in CTLA-4 blockade-treated patients, indirectly supporting the importance of the microbiome to patient outcomes. My feeling is the Zitvogel and Gajewski studies in some respects needed to happen in order for us to justify prospective studies of intervening upon the stool microbiota in humans. Next slide. So what about the limitations of these models? Next slide. Transplantable models, which dominate the preclinical checkpoint blockade field, variably recapitulate human disease. I will show just 4 examples, but there are several others in other disease types, such as lymphoma, breast, prostate, and other cancers. So of the transplantable models, the melanoma B16 model is really quite similar to human disease, in as much as it displays CDKN2A homozygous deletion. It harbors a very high number of non-synonymous single nucleotide variants. And in one study out of Uyghur Sahin's lab, 16 out of 50 expressed neoantigens were found to elicit a neoantigen-specific response. Advance, please. The ovarian ID8 cell line is somewhat similar. It's derived from a spontaneous in vitro malignant transformation of black 6 mouse ovarian surface epithelial cells, so its origin is similar to human disease. It does resemble serous ovarian cancer histologically and in tumor behavior. However, its sequence hasn't been published, so we're not sure the extent to which it resembles disease genetically. Next slide. The myeloma J588L line is really quite dissimilar to human disease in as much as it's used as a model for anti-PD-L1 efficacy, but at the same time this is one of the least checkpoint blockade sensitive diseases in human studies conducted to date. Next slide. Advance, thanks. So in colon cancer, 2 lines are commonly used, MC38s, which are quite dissimilar, and CT26, which are somewhat similar to human disease. MC38 is dissimilar in as much as it has a very high number of expressed single nucleotide variants or mutations, many more than is typically seen in human disease outside of mismatch repair deficient disease. CT26, however, has homozygously mutated KRAS. APC and P53 are not mutated, but CDKN2A is homozygously deleted. And interestingly, MHC class 1 but not class 2 is expressed. Next slide, please. As we increasingly rely on combination therapies, autoimmunity will continue to be a challenge to contend with. Interestingly, seminal papers on CTLA-4 and PD-1 did show that autoimmunity would be a problem, but the nature of it was somewhat different than that observed in humans, in particular the cardiac toxicity of receptor knockout mice, which thankfully has not been a problem in human studies. Furthermore, autoimmunity is infrequently observed and quite difficult to measure in mice. There's one study in which 3 pathways are targeted, trail CD40 and CD137, without adverse effects. That being said, certain strains are more prone to autoimmune side effects, for example, NOD and SJLJ mice, which might make a better context to study. As an example of this, ipilimumab induces hypophysitis in about 4% of patients. Iwama and colleagues used a mouse model to show that repeated injections of CTLA-4 blocking antibody in SJLJ or Black6 mice led to a lymphocytic infiltration of the pituitary and circulating anti-pituitary antibodies. Next slide. As I mentioned at the start of this talk, the homogeneity of our mouse models has facilitated great strides in studies of immunotherapy, but is somewhat of a double-edged sword. Through intentional inbreeding, the haplotypes of class 1 and 2 alleles in experimental mice are very limited, as shown in the upper left-hand panel. In contrast, the number of class 1 alleles in humans is in the thousands and rising as populations in additional geographical areas are sequenced. In fact, for a rainy day, check out the second website, allelefrequencies.net, which is a fascinating tale of how diverse humanity really is and how HLA typing tells a story of migration over history. In any case, advance please. HLA alleles are the tip of the iceberg. Other areas of diversity doubtless important to outcome include exposures, both environmental and therapeutic, the microbiome, as I mentioned, age, gender, and other factors. Next slide. Finally, one of the major issues contrasting between transplantable models and the human realities I alluded to briefly before is the time course of disease development. Schreiber's models have taught us so much. In the top half of the top panel, he shows the importance of rejection antigens, as I mentioned earlier, by using exome sequencing and neoantigen prediction. Nevertheless, these tumors are implanted for a mere 11 days before interventions start, including checkpoint blockade. Advance, please. In contrast, in a landmark study published now several years ago by Dr. Iacobuzio, her analysis of the rate of mutation development showed that even pancreatic cancers known for causing rapid demise among our patients probably develop over the course of years. To be fair, given their lifespan, the contrast from days to years is not quite as dramatic as if mice lived 50 times longer than they do. You can advance, please. That being said, undoubtedly the time course of the immune system's knowledge of an interaction with the tumor impacts the data we get from studying the tumor and tumor microenvironment. Next slide. So in conclusion, I hope that I've shown today that the proof of principle of the importance of checkpoint blockade and other immune-related pathways can be modeled in mice. Novel combination therapies, as well as combining modalities in different diseases, are under development based on these models. Next. Thank you. The extremes of toxicity can be gathered from knockout models, although the details tend to be different when antibodies are used in humans. Advance, please. And finally, predicting a disease-specific application is generally not realistic with many of the available mouse models. I thank you for your attention, and I'm happy to take any questions.
Thank you, Alex. That was a terrific talk. So why don't I open it up for questions, if anybody has any for Dr. Snyder? We might just need to make sure you have a microphone so that she can hear you.
So just out of interest, because that would help a lot in those kind of studies, for the 2 genetically modified melanoma models, why do you think one responded and one did not respond?
Do you mean the 2 I showed towards the beginning?
Yeah.
If I'm thinking of the right ones, with the blue and red lines, those were 2 sarcomas. And the first one, the KP and KPR derived, they only have 2 driver mutations. I think they've since been sequenced and they have a very low number of mutations, so there really just wasn't a lot for the mouse's immune system to recognize as foreign. This paper goes on to use an antigen to look at a number of different issues in the tumor and tumor microenvironment. In other words, the slide I showed is really just the introduction to the paper. And then in the second study, where there are thousands of mutations, there's more potential for the development of neoantigens that the immune-competent mouse can recognize.
Thanks. Any other questions? Okay. Thanks.